group, so I want to share with you initially how I got to work in that field and a little of my background. There's an additional burden upon me because I'm a local boy. And actually, I was trained in chemistry and physics, and basically I was also part of the MIT, Manila Institute of Technology, but it was really very ironic because this was in about 66 or 65 when we did the first experiment there, and it was actually 2 years before that I was on the roof of next building with a machine gun. And this is the only experience that an Israeli can go, that you are, within 2 years, you're on a machine gun on a roof next to it, because this time Jerusalem was divided, and within 2 years after that, you are really doing an experiment. And the first experiment was to isolate vitamin C. This is the first experiment you do that. Anyway, I finished my education here, and then I did PhD at the Weizmann Institute in sort of biophysics, spectroscopy with the late Isaac Steinberg. I was really fascinated with fluorescence spectroscopy and all that, and liked really to work on understanding basic mechanisms which have to do with biophysics and so on and so forth. And I was starting to think about postdoc, and I was starting to communicate with Luba Stryer of going to be a postdoc in his lab, and this was in the summer of '73. Little did I know that within 3 months I would be recruited to the Yom Kippur War, which was very long experience for me. I was mobilized for 6 months, and so imagine at the end of your PhD that you suddenly has to go to 6 months, you almost forget everything. And I had to cure myself from a very terrible event that took place in the Golan Heights. I was a captain, young 28 captain, which was leading 60 people. So it was really quite tough to get. I really didn't know what I want to do. But then there was a meeting, I think Daphne, you must have been in that meeting in Ayelet HaShahar in '74, in early '74, where a lot of people came from all over the world. And I started to develop, again, interest in science. And I met there a young guy called Elliot Elson, who worked with Watt Webb, and they developed the method of fluorescence correlation spectroscopy, which is kind of famous today. I was really amazed by the fact that you can use noise to detect single molecules, measure their diffusions, and study interaction. I thought I want to do postdoc there. And he accepted me immediately. This was those days, you meet someone, he likes you, and accepted it immediately. And I arrived to Cornell University with the ideas of using biophysics. Now I arrived there and I realized it was in the physics department. I couldn't do anything better than the local people in terms of the methodology. The idea was that I will translate it to doing experiments with cells using microscope. And I worked with 2 very capable guys, and this did not work for several reasons. The first one is a scientific one. The computers were very slow, so when you illuminate a laser, the cell will move by the time you average the data. So this would work. But there was another one which was even more trivial. It was a sophisticated physicist who worked there, and we were studying cells, and no one knew anything about the cells. So they asked me, do I know? So I said, well, you guys know less. So I started to grow cells, which we will get from other labs, and I noticed that they're really changing. And I became quite good friend of F. Rucker, who was seen here in the picture, and I said, F, what is this? So I said, idiot, you are now growing yeast. So I felt that I'm tremendously ignorant, and I said to Elliot and Watt Webb, look, we have to solve this problem. We have to solve this problem. I have to learn how to. I realized that I cannot contribute to the physics. They know what they're doing. So I took myself and I went to a Cold Spring Harbor course on cell biology. And this is, you can see how things change in life. In that Cold Spring Harbor, there were some students, and one of my roommate was someone called Graham Carpenter. So in parallel, during those days, you will go to the library and read literature that you're not supposed to read or did not expect. You really go and browse. And I started to realize that there's some interest in the field of growth factors. There was an old paper by Howard Temin, which shows that cancer cells need less growth factors or serum because they make perhaps those. And then Graham Carpenter introduced me to EGF, And I thought this is kind of interesting. And he said, why don't you add EGF to cells and see whether the diffusion changes? So he gave me like 5 milligrams of EGF. I did experiment, nothing happened, so I had 5 milligrams of lyophilized EGF, which was a quarter of the world's supply. And so basically, now you will see how things started. I was measuring diffusion, and basically I thought that it's really better that I start to do some biology and study about some receptors. So the first lecture, I'm going to tell you about receptor tyrosine kinase, a brief introduction, a zipper code mechanism for RTKs are activated, and the discovery of kinase inhibitor treating cancer. But before that, this is the sort of mechanism that we thought receptors work. And I think Bob will recognize this mechanism because he probably had a similar one at that time. There's an EGF molecule, and it binds to a receptor which activates something. And the literature, all the knowledge in the literature at that time was the following: that NGF and EGF were already discovered by Rita Levi-Montalcini and Stanley Cohen. This is very important. This was a key paper that made me interested. This is not some of the work that is known for Howard Temin, that cancer cells need less growth factors for proliferation. Then certain cancer cells produce TGF-alpha, which is another ligand. And finally, they act on surface receptors. So I said, well, I'm expert on receptor diffusion, maybe I'll start to look on it. And there was some more information that EGF bind to EGF receptor with 2 affinities. Later on, many years shown due to negative cooperativity. Later on, I'm not yet there, it has intrinsic tyrosine kinase, and there is internalization. So I decided, I said, well, it's very similar thing that came in the theme earlier. In order to understand them, I have to purify them, and I have to clone them. At that time, I didn't know anything about cloning. So I said to Elliot, I want to purify these molecules and sequence it. So he said, no, you should do recombinant DNA. You use cDNA. So I started to read about cDNA, how you can do it. But then how would you purify a molecule that you don't have any way to interact with it? So we used a different approach, which was very effective. This was the time that the monoclonal technology came. So I came up with a scheme in which we take membranes for cells which have a EGF receptor and screen from antibodies which block ligand binding. And those which block ligand binding can some of them be used for affinity chromatography. And this is what I started to do when I first came to Israel to be a faculty at the Weizmann Institute. And one of the 3 people worked in my lab on this project, Yossi Adan and Tuvia Lieberman and Irit Lacks. And after several years, this came up. We really purified EGF. It was very difficult. No other lab could do it. And then, of course, we were able to get partial sequence with Mike Alderford, and everything changed. So this was the second example where an oncogene was shown to be an active. The first example of oncogene was shown to be an activated receptor. So even from the primary structure, we can guess such a mechanism. And then, of course, we collaborated very quickly with Axel Ulrich, who there are very few people that can clone, and we very quickly cloned the receptor. So it was interesting case here that you have here an activated receptor, which has a ligand binding domain, a kinase domain, and a phosphorylated tail, and then a truncated receptor, which is oncogenic. And this was very interesting, and this started to establish a pathway, because a year before that, the cis oncogene was found to be a PDGF. So you have a ligand that is an oncogene, you have a receptor which is an oncogene upon activation, and then you have also RAS. So this was already thought at that time. Now, is it relevant? So it was an oncogene in avian cancer, which I'm not showing to you. But very quickly we realized already in that time, and this is now early 80s, that certain human cancer, like glioblastomas, overexpress this gene. And there was also gene amplification. You can see these are all very, very experimental. So there was an interesting link in cancer. Already at that stage, because we had monoclonal antibody, I said, well, if cancer is driven by this mechanism, maybe this is a good way to develop new drugs. But of course, I was really far away from this. So how does it really work? the EGF receptor is a tyrosine kinase. What did we do in these old days? We would stimulate cells with EGF and then blot with phosphotyrosine antibodies. At that stage, of course, Tony and I started to see each other very frequently, and you can see very nice. This is one of the first experiments in the area for protein. It became also quite clear that the best substrate of EGF's activation is the receptor itself. And then we started to look on different substrates. So that meeting in Titisee, where we described it, there was someone called Suguri in that meeting, worked with Errol Studman. He, at that meeting, described the cloning of phospholipase C, which also had an SH2 domain. So this started to make sense. So he gave me and Tony aliquots of anti-phospholipase C, and we did the following experiment. So what you take cells, you treat them with EGF, immunoprecipitate with phospholipase C antibodies, and immunoblot with phosphotyrosine, and you see two bands. So what are the two bands? One of the bands is EGF receptor, one of the bands is PLC gamma, and one of the bands is EGF receptor. So not only that EGF phosphorylated PLC gamma, they formed a physical complex. So we started to realize something interesting is happening, because at that time, SH2 domains were already described, and just about that time, Hanafusa showed for the first time that SH2 domains can bind phosphotyrosine. So we started to think that probably they make a complex through an SH2 domain. So this is indeed what we published. There were two papers at that time came in Cell, which basically shows that SH2 domains of PLC gamma interacts with the phosphorylated tail of EGF receptor, and this leads to activation of 2-second messenger and to phosphorylation of PLC gamma, which is then activated. So you have, so it does two things, but then this was one of the key messages, that the targets of receptor tyrosine kinase, unlike G proteins, here the receptor contains all the information for the signal, the ligand binding and the kinase domain and the regulatory elements are in the same very molecules. You don't need anything else. So then we decided that maybe if this is really taking place, why don't we use it for how to clone new targets? So I came up with an idea that since at that stage we could prepare large quantities, maybe we take this region and we do expression cloning and find new targets. So everyone who knew anything about expression cloning told me it cannot be done. And then two nephrologists came to work with me, Ben Margolis and Ed Skolnick, and I told them and said, great idea. You see, they were so ignorant, but they liked it, which is sometimes is an asset. And indeed, so this is what we did. When one of them noticed that there is a methionine here, so he used a cyanogen bromide, and he prepared very hot and radioactive, which was possible those days. Now one doesn't want to touch it. And we started to use expression libraries. And this turned out to be extremely productive. We cloned a lot of SH2, some new, some proteins. So this is the approach. You really cleave the tail of the receptor, and you make it extremely radioactive, and then you express. And then this is one of these experiments. You see, this is one of the spots, and then you really purify and draw it, so you have. So we started to call this molecule growth factor bound, receptor bound, GRABs. So some of them are kind of famous this day. And I show you the first fishing expedition. So this way actually how PL3 kinase was cloned, because it has an SH2 domain. So it was a very productive time of the lab. And then of course, second molecule called GRAB2, which became an important linker. And then of course you see the list of molecules. So when we cloned so many molecules, and all of them had SH2 domains, so we were really busy to see what is going on. And I'll show you just one or two examples. And this started to really establish a mechanism how signal is propagated. That the receptors are not only enzyme, but they are also a platform for recruitment of signaling molecules. And this is basically showing you some of those. So this is all the different enzymes which have been recruited by receptor tyrosine kinase. SARC, so an SH2 domain, PLC gamma, and many other here. And so the idea is that SH2 domain is being recruited, and then this target becomes phosphorylated, and this links to signal activation. And I'll show you these other molecules which were interesting, because these molecules contains only SH2 domains, only binding motifs. So it very clearly, you can really think about them as adapters, that they will use an SH2 domain to interact with activated receptor and an SH3 domain to activate with something else. And then a paper came from David Baltimore lab, and we used to meet quite frequently because he was at the Rockefeller, which shows that SH3 domain bind to proline-rich sequences. So this was already telling us how these signal pathways are being activated. And basically, this is SH2, this is GRAB2, has an SH2 domain and two SH3 domains. And so there was a lot of evidence to come up with the following mechanism. But before I go and show you how this works, and this was done in the very early 90s, so this is comparison of EGF receptor, PDGF receptor. There are enzymes that phosphorylate many other substrates, but the best substrate of each receptor is itself, because this is the mechanism how signal is transmitted. They provide a platform for recruitment of molecule through unique phosphotyrosine motifs, which are the targets. So you can see how you assemble a large complex which propagates the signal. And so, and basically, it was shown, and I don't show his structure, that SH2 domain interact with phosphotyrosine moieties, which is the major binder, together with nearby few amino acids. So specificity is determined by nearby amino acid and phosphotyrosine binding. So this is how specificity measured. And then it's also indicated that SH3 domain recognize similar motif of XP, XSP, which appears in different targets. So it was quite easy to compare genetic pathway which emerged from Drosophila and nematode genetics with this biochemistry. So then in 93, we were for the first time able to link how receptor tyrosine kinase are activating GRAS. So there was a lot of evidence at that time that they are in the same pathway, and there's a lot of genetics evidence, but this was the mechanism. GRAB2 is bound to proline-rich in the exchange factors for RAS called SOS. Then through, when it's activated, when a receptor is activated, it interacts with particular sequence here, which involves this motif, brings it to the membrane, and this is how RAS is activation. So this is a simplified mechanism, but in a nutshell. So you don't need here the G proteins, because the activity which transmit the signal is part for the same receptor. Cytokine receptor somehow involved that the kinase activity is separated. So it's an interesting evolution of how that happened. So receptor tyrosine kinase in the 90s, if you need to remember what are the key experiments, SH2 domains were identified by Tony Paulson, and then Hanafusa, both of them unfortunately are not with us, interact with phosphotyrosine, and then SH3 domains bind with proline-rich sequences. The adapter protein GRAB2, composed of SH2 domain flanked by two SH3 domains, interacts with receptor tyrosine kinase, leading to MAP kinase activation. And finally, receptor tyrosine kinase function both as enzyme and platforms for recruitment and amplification and of signaling pathway. So these are the general concept. And everything beyond that are details, extremely important details, but this is how the signal. Of course, this is a very simplified view, but this is what I can do now to describe the history in a short way. Now, subsequently, and I mentioned the similarity to the receptor U, Brian, when I saw this scheme, which is very similar to what you were doing. There are other motifs which interact with multiple proteins. So you can see here how these different motifs can really generate a really complicated network of interactions which provide a lot of different signals. So it's really complicated story now, and this is basically what people are telling me now. It doesn't matter what you see here. This is one of these networks. There are multiple reactions. The people are using system biology, all different type of kinetic parameters. This is what you start to find out. But it's all governed by this simple concept. And I think everything beyond that is just extremely important details, but these are the conceptual aspects. There was another topic which was important to figure out. And at that time, we already knew that there are 89 protein tyrosine kinases. 58 of them were receptors and 31 non-receptors. And this is the largest group of dominant oncogenic proteins. So this was early 90s And it became clear that it would be very good to try to develop some ways to develop drugs based on that. And so this is a scheme of all receptor tyrosine kinase, which was established before the Genome Project. And yes, you can see the hallmark of this receptor are, with the exception of insulin receptor, which is a dimer, that there is a large extracellular domain separated by a single transparent domain which interact with which has part of the tyrosine kinase domain, which is a transmitting signal and then regulatory sites. So there was a question here, how is the signal transmitted? And early days, one of our monoclonal antibodies was able to activate EGF receptor. And when we made FAB, so I made the prediction that it's activated by dimerization. And it sort of makes sense because if you can really bring them together, I received a lot of criticism about it during the years. And it was very difficult to really convince people that this is the case. Of course, it's like there are 3 stages of truth in science. First, it's not true. Second one, it's not important. Third one, we knew it all along. So this is the case. So it turned out that this is a general mechanism because Axel Ulrich and I in '86 did the following experiment in which we really created a receptor which involves extracellular domain, swapping the extracellular domain of the kinases and all that. So this is the EGF receptor and this is the insulin receptor. So here you have EGF receptor outside, insulin receptor inside, and vice versa. So when you add insulin, you get EGF responses. When you get EGF, you get insulin responses. So extracellular domain determines ligand specificity. Cytoplasmic domain determines physiological response. So what was this pathway? So the mechanism of activation, which I spent a lot of time studying, is that receptor tyrosine kinases are activated by receptor dimerization or by higher oligomerization. And I'll start to tell you some details about few receptors because it's a very broad field, so I try to sort of give you information about conceptual think. And this is the sort of latest stage of the receptor, and there's more evidence that the concept that you guys described of bias and effects of kinetics and different states exist here, and I've started to show you some of that. Basically, this receptor exists mostly as inactive monomers, which are in equilibrium with dimers, which are either inactive, or activated. The function of the ligand is to induce the formation of an activated state and also activate it once you have an allosteric transition. And this is an important equilibrium. In some cases, there's evidence that higher oligomerization state. We are starting to get evidence that there are different states of activated receptor, and there are some kinetic aspect. It's more difficult to study them considering that these are really 2 separate entities, but we do have evidence. So how is mechanism of activation of receptor tyrosine kinases mediated? Some of it simply because the ligands are dimeric. So a dimeric ligand will bring to the receptors, and these are a few examples. Another one, you have a ligand-induced receptor-mediated dimerization. These are EGF receptors. I'll show you shortly cartoons from structure. The third one is insulin receptor, which is a pre-existing dimer, but in an inactive state. So what the ligand does induces a structural change which juxtaposes the catalytic domain. And finally, sometimes you need more than 1 for tango. You need 2. FGFs, which I'm going to describe in some details, they alone do not activate FGF receptor. They need heparin sulfate proteoglycans. So this is the variation of the theme. So after purifying the receptor, we started to embark on understanding this molecular mechanism, the structural biology. And in the next slide, you see schemes of some of the structure we solved. These are really scheme based on structures. So you can see here that always you have 2. And so this is the kit, which I'm going to show you in some detail. The ligand is dimeric. And this is EGF receptor, 2 ligands induce receptor dimerization. This is angiogen receptor. Interface is formed by the ligand itself. And this is FGF receptor, where you have heparin and an FGF to add together. But you always induce this response. And so I'm going to focus the attention and describe you some details on stem cell factor receptor as an example and kit. So stem cell factor receptor has 5 IgG domains in the extracellular domain and a kinase domain and signaling. It's activated by a ligand called stem cell factor, and there's a family of 5 receptors which have 2 different ligands which are activating this mode of action. So a little introduction about this ligand. What is stem cell factor? Stem cell function as a homodimer. Each protomer contains 161 amino acids. It exists as both membrane anchored and soluble isoforms. It's required for development of hematopoietic cells, melanocytes, germ cells, and intestinal pacemaker cells. And so this is how the ligand is activated. And so if you knock out the receptor, it's embryonic lethal. But if you have mutations, which is a weaker mutation, only one in the allele, it's negative. It really has a dominant negative effect, which leads to this pigmentation, which is very similar in mice and man, which also shows you the fidelity. So if you don't have enough of a signal activated by KIT, you have developmental problem. If you knock out both allele, it's lethal, but then you have this, which actually is another verification for the dimeric model because it's a dominant interfering dimer. So this is when you don't have enough signal. If you have too much signal, it leads to various cancers. So activated KIT mutations exist in gastrointestinal stromal tumors, in AML, in melanoma, about 5% of melanoma, in mast cell leukemia, and idiotypic fibrosis, and even in CML. So there's a lot of cancer, so this is why it's important to block this kind of activity. So we spent a lot of time understanding how these molecules work, and then in 2007 we had for the first time a crystal structure of the ligand alone, of the extracellular domain entirely, and then free or occupied by the ligand. So these are the crystal structure. So we can really come up with a model how this receptor is activated, and I've animated it for you. So this is the extracellular ligand binding domain. It has 5 domains, D1, D2, D3, D4, D5. This 3 one encompasses the ligand binding, which is a dimer, and then there are 2 very flexible hinges which connect D4 to D3 and D5 to D4. You can imagine that there is a transmembrane domain here and kinase domain inside the cell. When the ligand binds, and because of this really flexibility of this hinge region, it brings the 2 receptors together, and then they form homotypic contacts between 2 D4 molecules. And these homotypic contacts are essentially required for receptor activation. And I'll show you the evidence for that. So you look on these D4-D4 interactions, they are mediated by 2 salt bridges, in which an arginine molecule from one molecule interacts with glutamic acid to constitute such a motif. So if they dimerize, but they cannot form these contacts by mutation, they cannot be activated. And this is basically shown in these slides, where this motif is retained in KITs for all species and in all members of the receptor family and in other receptors. So this constitutes a dimerization motif that when blocked, the receptor still can dimerize, but they cannot activate because these contacts cannot really occur. So this is how the receptor transmits the signal to bring 2 cytoplasmic domains together. So this was one indication from the structure that it's how it's mediated. Second one, and this basically shows you here. So this is tyrosine phosphorylation of wild type receptor, and then when you make this point mutation here, even at very high concentration, and dimerization is absolutely normal. So another indication was as follows. This is the transmembrane domain. This interacts in D4. This is this D5 region. A lot of oncogenic mutation occur in this region, where at the interface between the small molecules. So we speculated at the time that this is what really leads to activity because it forms constitutive activation. I'll show you shortly that this is proven by structure, and we know exactly the strengths of the affinity that is mediated by this change. And this is basically how this receptor, I think, are activated. So the ligand is dimeric, or some other ligands induce dimerization with different mechanisms. They come together because of the flexibility of these hinges. These regions come together, link 2 cytoplasmic domain together. There's tyrosine phosphorylation, which occurs in trans, and further activation. Now, so a few years later, before we started to use cryo-EM, we did some negative staining. If you want to see the structure of intact receptor, this was done like 5 years ago. We are doing a lot of cryo-EM, and basically these are images of individual and combination of averages of full-size receptor. Here you see the crystal structure, high resolution, and here you see basically a full-size detergent solubilized receptor. You can see that it's already see very clearly here, even in these 50 averages, that it's very similar. So you can really collect. This has been published, and you can see that we can really get two forms of receptor in relatively good views involving thousands of averages. And the nice thing about this is that you have different states, and at that stage we already started to think that the possibility the different states induce different elements. So these forms, we can really look in some details because we had very good data. So we can really have two forms of this receptor, and so moreover we have the crystal structure of all of all parts of the molecule at high resolution. And basically what is remarkable about this is that the extracellular domains is always very symmetric, but there are different arrangements of the cytodomain. They are always asymmetric, as if the kinase domain can afford. And of course we want to have a high resolution data to really see it, but in the meantime we can dock into these structures of the currently available structure, and you can see that it fits quite well. We don't know how this interacts exactly here, but of course you want to do a high resolution, but this is how it looks like. We also don't know how the transmembrane domain, but this is a view of how these receptors look like. It fits very well with the extracellular domain. Now let's look now on this oncogenic mutation. So it would be nice to see how they really make this receptor oncogenic and active. And so oncogenic activation of KIT occurs by four hotspots of mutations. A mutation in the kinase domain, which stimulate kinase activity, leads to constitutive activation of the kinase. A mutation in the juxtamembranous region, which releases an autoinhibitory constraint, and two hotspots at the extracellular domain, which involve different types of mutations. And these are the mutations which I showed you. So this has been established by structural analysis, so we decided to have a look at this. And in order to do this, we made all the different forms of these mutants in domain D4 and 5. There was no need to make the entire molecule. So these are all the different mutations. And some of them made our life easier because they were not expressed. Other one expressed but didn't crystallize. One mutant here made beautiful crystals which didn't diffract, and second one made very ugly crystals which diffract very well, which reminds me, as what my mother told me, always look for internal beauty. So this one had an internal beauty. This was really misleading. So this has been, the crystal structure was determined, and the interactions, this is for the structural biology, interaction between these regions are determined by the crystal packing, and this is the relevant part that we have looked into it, and it really shows you very nicely how a normal receptor becomes an oncogenic molecule. And we decided to study it in great detail. And basically we made a lot of mutations here, and we really measured the affinities to understand what sets a normal regulated ligand-dependent receptor from an oncogenic molecule that becomes activated by a single bond mutation. And it's a very simple answer. Change in affinity of this region here. And this basically was published a few years ago, and it basically shows you that the strength and cooperativity of KIT ectodomain contacts determine normal ligand-dependent stimulation or oncogenic activation of cancer. This is basically what it is. If you have a mutation which forms a constant association, this is what it goes. And basically the affinity of this interaction. And I'll show you the evidence for that. So this is what we did. We took these two domains and we measured their binding affinities, and then we all remember these mutations which really is required for activation. For the wild type receptor, if you mutate this critical salt bridge, you can barely activate the receptor. but the affinity is really very weak, barely detectable. You cannot measure such affinity. So the affinity that take place here are very weak, but this is why I call it a zipper-like mechanism. But look what happens when you have this constitutively activated receptor. Well, this is just summarizing. So in the case of ligand-dependent KIT, there's weak binding affinity of isolated D4 and D5 domains, and ligand-induced KIT stimulation is dependent on the salt bridge. So this is how it's normally regulated. Weak affinity and sensitivity. Okay. This is what it takes to be a normal ligand-dependent receptor. Now look at this constitutively activated receptor which drives cancer. It's already ligand independent, and the ligand does not further increase the affinity. And look, the affinity is increased by 200-fold. This is all what it takes for a molecule to be activated. When you really make this salt bridge, it doesn't really matter. It's already activated through this interaction. So change of 100-fold affinity and leads to constitutively activated dimeric receptor, which lost the ability to be regulated by the dimer. But there is an intermediate state. There are some other mutations, and this just summarizes the binding affinity. This is constitutively activated ligand-independent oncogenic mutation. The ligand binding affinity is 200 to 500-fold. We don't know exactly the number. We have precise value of the mutant, but we don't have precise value of the normal because it's so weak. So this is an estimate. And then ligand-independent activation does not depend upon salt bridge, which is required for activation. But there is another oncogenic mutation, which is what we call sensitized receptor, where the affinity is only about 10 to 20-fold. Here you see you have basal activity, and the ligand further stimulates it, and here the salt bridge is essential. Okay? So there are different grades of oncogenic activation, some totally activated and some what we call sensitized. This oncogenic mutations requires ligand in order to uses the local ligand. And this is summarized here. So ligand sensitized oncogenic KIT mutation, the binding affinity is increased by 10 to 20-fold. Increased basal kinase activity that is further enhanced by ligand stimulation, and kinase activity depends upon salt bridge. So these are the stages, and of course you can find similar thing in other receptors. So based on that, we propose this zipper-like mechanism for RTK activations, which basically this molecule evolved to be monomeric, and they do not interact, and then they have the capacity of weak interaction along the entire molecule. And these interactions are not sufficient to make stable dimers, which are activated unless you bring a dimeric ligand, which increases the local concentration. And then because of the flexibility of the hinge, this weak affinity, which are very weak, act in a zipper to create this sort of state of activated. Now in some oncogenic mutation, which increase the affinity only by 200-fold, you don't need any of that. And some of them you can really use more effect. So this is a zipper-like mechanism, and elements of this pathway really can be detected in all receptor tyrosine kinases. So in the 90s, I paid attention to the possibility that one can really develop drugs in this field. But protein kinases were not thought to be good drug targets by pharmaceutical industry in the mid-90s. And this is the reason. And we received a lot of criticism. Difficulty in targeting intracellular enzymes and tyrosine kinase domains of receptor tyrosine kinase. They have to enter the cell. This was one criticism we received. Difficulty in obtaining the requisite specificity due to the similarities in the ATP binding pocket of protein kinase and many other ATP proteins. These are all arguments that we heard at that time. Difficulty in obtaining the necessary binding affinity to compete with tight binding and very high concentration of intracellular ATP. And so it was very difficult to prove that. But of course, now we know that this is really not the case. And these are now the protein kinase inhibitors which exist. There's 45 FDA-approved protein kinase inhibitors, 5 FDA-approved therapeutic antibodies targeting receptor tyrosine kinase or their ligands, and more than 400 kinase inhibitors are in clinical trials, and numerous therapeutic antibodies are now in clinical trials. So contribution of my work to this is as follows. So this is the drug discovery activity that came from my lab and my colleagues. The first company, this was the first biotech company to develop small molecule kinase inhibitors for cancer therapy, were founded by Axel Ulrich and myself. S stands for me and U for Ulrich, and it was founded in 1999. And there are 2 FDA-approved drugs which came from this work. Subsequently, I founded with other colleagues Plexicon, which is, I became very much involved in structural biology, and I thought that structure-based designed kinase inhibitor could be productive, and this turned out to be an extremely powerful approach. And there's one FDA-approved drug, the second drug now in phase 3. And this was the first melanoma drug for BRAF, which was developed by Plexicon. At certain stage, I realized that I made a mistake by focusing only on small inhibitors, and there's a lot to be done on using therapeutic antibodies. For example, we have a therapeutic anti-KIT, which is 1,000-fold more potent than Gleevec. So I founded Clotho, and eventually it merged with another company called Celldex. And so there are 2 drugs in clinical trials, which I'll show you one of them. It works an interesting allosteric mechanism. And then, of course, recently I started another company, which is a totally different story. I never yet spoke about it. It's some kind of enzyme replacement therapy for calcification. It's an interesting science, but it's not part of this, and I will not discuss it. So these are the 3 drugs that came from companies I founded. And of course, we know the advantages of these drugs, and we know the limitations. No treatment of that type is a cure, and there is a lot of interest in developing ways to address the resistance. And I'm not involved in that, but this is where we are in this field. So there are also 2 therapeutic antibodies. One of them is a very high affinity, really potent anti-KIT inhibitor, which is 1,000-fold more potent than Gleevec. And it's very effective from diseases which involve overexpression of KIT. It's now in clinical development. And then I'll show you, before I finish this part, is an interesting allosteric inhibitor. It's an RB3 antibody, which is now in clinical trial for certain cells. What I really like is how we resolve the mechanism of how this molecule works. And it shows you that you sometimes can take a potential drug and elucidate something very interesting mechanistic about it. So I have to tell you about how members of the EGF receptor family are activated, which I spent about 20 years studying. So in the case of EGF receptors, members of the receptor, there are 4 members of the family: EGF receptor, RB2, RB3, and RB4. EGF receptor has ligand binding and kinase activity. RB2 has no known ligand. So far, maybe not, but has a very strong kinase activity. RB3 has a ligand, but it doesn't have kinase activity. It's a pseudokinase. And RB4 is like, it has ligand and so forth. So each of them can function as some kind of homodimer by combinatorial heterodimers. So it's a very rich repertoire of signals because they can stimulate responses, primarily all those with activity as homodimers, but they also form heterodimers. And I'll give you the reason why. And there are really inhibitors for EGF receptor and RB2, which are in the clinic, quite successful with all the limitation of small inhibitors. Here, there's a lot of evidence of playing a role in cancer, yet you cannot block the kinase activity because it doesn't have kinase activity. So there are a lot of interest in it because of the following reason. And this shows you how members of the EGF receptor are activated. You have an inactive state, which is held by intramolecular interactions, which exist in an equilibrium with a ligand binding state, in which 2 regions in the receptor, what we call 1 and 3, had to come together to really lead to receptor activation. And the receptor activation is mediated by receptor interaction. It's perhaps easier to see it by this cartoon. So the ligand binding constitute domain 1 and 3, but they're far apart in an inactive state, which is held by these intramolecular interactions. But this is a reaction in equilibrium. And then when the ligand binds, it traps binds the 2 domains together, and then this region here interacts to form heterodimers. Okay? So this is how it's equilibrium between an inactive autoinhibited state and open state poised to form heterodimers, in which 2 regions here bind to ligand binding. Now, RB3 is really going through the same process. But why is ERBB3 important? Being a member of the EGF receptor family, it's expressed in epithelial cells, nerve tissue, and a variety of cancers. It's activated by neuroregulin family of ligands. And unlike other members of the EGF receptor, it's a pseudokinase. It doesn't have kinase activity. More information. ERBB contains multiple PI3 kinase sites, a strong activator. So it's a strong cell survival pathway. It forms heterodimers with activated receptor. It excludes PI3 kinase and all that. And it was proposed that ERBB3 functions as an obligate heterodimer of EGF receptor, ERBB2, or ERBB3 through heterodimer formation. And finally, ERBB3 signaling is an important driver of tumor survival and proliferation, as well as poor patient prognosis. There's a lot of genetic evidence that suggests that case. So there are two different cancers and mechanisms how ERBB3 plays a role in cancer. And you will see how one structure revealed how it happens. What we call ligand-stimulated manner. By ligand-induced stimulation of heterodimers. Very simple mechanism. But there is another mechanism in which is called a ligand-independent manner. Many tumors overexpress ERBB2. By virtue of the overexpression, it shifts the equilibrium and it interacts with ERBB3, and then this provides a survival coup by recruiting multiple pathways. And some cancers really utilize primarily this process, and some cancers this one. And then there's a third rare mechanism in which there are oncogenic mutations which relieve this autoinhibition. So these are the three mechanisms. Now, this is how much interest there is in the drug company to develop this. So this is the crystal structure of inactive ERBB3, which I've shown you in this inactive state. Ligand binding constitutes interaction with these two domains, but they're far apart here. So these are all the different companies which have ligand binder antagonists, and most of them have been directed against blocking ligand binding. So there's no way of thinking how they really block the ligand-independent mechanisms. So these are the different drugs, and this is how it really maps to. Now, so we took an FAB of one of those, and this is related to a antibody in Clotho, the company I co-founded, and I have financial interest in that company, and we solved the structure. And initially, it was not clear how it works, because look, these are the regions where the ligand binds, and look, the antibody binds to totally different area here. So it blocks ligand binding, but it really doesn't interact with the ligand binding domain. But when you look at the structure, and first of all, you can say it through an animation here, you can see that very quickly that it binds to an area which is essential for this release of autoinhibition. And I will show that to you. So you see, so the ligand binding is this region and this region, and you see it blocks, you see this is in yellow where the ligand, it doesn't interfere with ligand binding. And this is what it is. So this is the area connecting domain 3 and domain 2, so it provides a wedge there. And therefore, it locks the receptor in an inactive state and does not allow this conformation and change to occur. And this is basically the scheme which I showed you. And this is where it really binds. And you can see how it provides a very nice wedge. And so it prevents this transition to an activated state. And therefore, it will block the cancers which are ligand independent and ligand dependent. So in one antibody, you can really do both, because for ligand dependence, they need to come together. But if they will never go there. So I am animated this to you so you can see it in a more clear way. So this is how it really normally occurs. This is this open and activated state. And so what happens is the ligand binds, and then of course they interact through this interface. What happens is that because of binding to this wedge, this is what happens. So it can rotate. So you cannot generate a binding site which would allow ligand binding. So it interferes with ligand binding through an allosteric mechanism and prevents heterodimerization. And just shows you side by side. So there are quite a lot of interest in blocking these receptors, and so far there is the most common way is inhibition of ligand binding. There are some antibodies which are in the clinic which block receptor dimerization, and this is an interesting new way of blocking receptor in an auto-inhibited configuration. So now I change gear and I tell you the real stuff, the more recent stuff. And I'll tell you new stories about endocrine FGFs, and actually that we can really capture an activated state of FGF receptor. And then I'll show you an interesting story that may be related to what we heard 2 days ago, in which you have an evolution of a sugar-cutting enzyme to become a receptor for hormone which regulates sugar level. How does it happen? And I'll show you a zip code mechanism for endocrine FGF signaling and new opportunity for drug discovery in metabolic diseases. So I don't think that many of you know much about canonical and endocrine FGFs and how they work. So this is the largest family of growth factors, and they fall into 3 categories. Canonical FGFs, and there is another one which is called which are called intracellular FGFs. It's very difficult to describe them because not much is known. And then there's 3 molecules which are called FGF 20, 19, 21, and 23, which are really endocrine hormone. Canonical FGFs act primarily by working together with heparin sulfate proteoglycans. And endocrine FGFs use a protein called Clotho. And I'm going to describe to you how they work. And they all activate members of the FGF receptor family, which are here, which compose 3 IgG domains and a cytoplasmic domain. Now, this is an interesting physiological system, because this is an example of a single gene that encodes for 2 receptors with two different ligands with 2 different affinities. So the extracellular domains of FGF receptor are composed of 3 IgG domains, in which the 3rd IgG domains is encoded by 2 alternative exons, a common exon and an alternate exon. And one of them is called the B isoform, and second one is called the C isoform. And interestingly, all B isoforms are expressed in epithelium, and they bind FGF3, FGF7, and FGF10, where the receptors are expressed in mesenchymal cell, and this is exactly opposite for the C isoforms, which are expressed in mesenchymal cells and are stimulated by ligands which are bound in epithelium. So this is an interesting system where a receptor, a gene of a receptor can encode for 2 different receptors with 2 different specificities. And sometimes it's mixed up in cancer, so you can really create a lot of different problems. Now, the reason why I saw that, because this Clotho protein only interacts with the C isoforms. Now, there's other things which set FGF receptor apart from other receptor tyrosine kinase. FGF binds to FGF receptor with a KD of approximately only micromolar, which is at least 1,000-fold weaker than EGF or PDGF binding to the receptor. It's very weak affinity. Why is that? Its stability of FGF receptor complex is greatly enhanced by interaction between a heparin sulfate proteoglycans or heparin. So this is offset by interaction with heparin to generate extremely potent binding. And so this is how it's maintained in an inactive state. So I mentioned that. So this is how FGF receptors are maintained in an inactive state. There is some region here which interacts with an acidic box, and the domain 1 occludes ligand binding, and there is an equilibrium similar to what we have seen in EGF receptor, but it's an equilibrium. So we again decided to solve the structure, and this is a structure we described already almost 20 years ago of a ternary complex, in which you see a heparin molecule, an FGF molecule, and a receptor molecule. And without domain 1 because it was flexible, so you cannot really show crystal. And this was thought to be, this is in textbook and all, thought to be a structure of an activated receptor. This was the first one whose structure was solved. Now, if you remember the scheme which I showed you, so this was the first molecule that was solved. And we didn't pay attention, we didn't notice how important it is that these regions here are so far apart. You see, always they're very close. And the question, I started to ask the question whether this is really the active state. And then we realized that there are a variety of disease or cancer-causing mutations in which there are cysteine residues which exist in a region which connecting the extracellular domain to the transmembrane domain. You see a new cysteine here, which is oncogenic gain of function. And actually, in all 4 receptors, you can basically show that this tyrosine residue is replaced by cysteine. So, and they're all activated. So if you have a cysteine so close to the membrane, it cannot be far apart for the receptor to be activated. So it started to test them and also looked into all the published crystal structure by us and others, and we realized that the crystal packing was such that 2 receptors interacted this way, and there's no way for them to crystallize in a way which will show contact. And everyone who published repeated our structure agreed with this, but it was not, it was something which was mixed up, and this was the first one. So I decided we have to really see what is going on here. And we were guided by these cysteines. It must be very close if there is a cysteine which leads to activation. And basically we analyzed them, and it turned out to be that this cysteine really can activate the receptor in 3 different ways. There is an activated ligand-independent FGF receptor 3, where you see here. Where you see here. So you see, this is normal receptor, and this is the 2 cysteines. You cannot stimulate them anymore with the ligand, and there are many of them make constitutive dimer. There's another form, which is called an activated receptor dimer that can be further activated. So within this cysteine, you have another cysteine here. You can see this is wild-type receptor, how nice and stimulated. You can further activate it. So they're all gain-of-function mutations, but they act in complicated way. And then there is another receptor which is really super activated. So this is clearly the case. So what does it tell us? So basically, we saw the crystal structure of one of the receptors, which is called FGF receptor 4 recently, and it really behaves in total different crystal packing, and it shows this homotypic contact. And so clearly, we think now that the activated receptor is very similar to others, in which the homotypic contacts are very close to each other. And this is basically how it looks, because there's another extension of FGF which interacts with another protomer. So we think that what's going on here, that there are different states of equilibrium between the different receptors, and when you really have crystal packing, it can really trap it into something of that type, but something like that exists. And we have now good evidence that this is really a dynamic interaction, and we just captured a state which is poised perhaps to be activated, but not that. So this is how canonically FGF act. But now, what about endocrine FGFs? Again, so let's focus about endocrine FGFs. So canonical FGFs need heparin. Endocrine FGFs need interact with Clotho. So first of all, what are endocrine FGFs? Normal FGFs have this cyan region, which I showed you in the structure, which binds to the receptor. All the endocrine FGFs have this extra tail, which renders it canonic FGF. It binds to Clotho molecules, and some of them are mutated in diseases, and like severe disorders which call bone disorders, such as FGF23. Now, a little about the biology. FGF19 is expressed in the intestines. It acts in the liver by activating a beta-Clotho FGF receptor Clotho, and as Charles mentioned, it is a major regulator of bile acid lipogenesis in the liver. FGF21 interacts also with FGF receptor 4, but with another FGF receptor 1c. It's made in the liver, but it acts in the hypothalamus and adipocytes, and it's a major regulator of energy expenditure. It's a hormone which is induced by a variety of stresses and also during periods of starvation. It's a starvation hormone. But it utilizes beta-Clotho and different members of the FGF. And then FGF23, which I'm not describing in great detail, it's made in the bone, it acts in the kidney and parathyroid, and it acts Clotho alpha with all members of the receptor. It's a major regulator of phosphate homeostasis. So David Mangelsdorf from UT Southwestern spent a lot of time studying the biology. I'll describe you a little the biology on what were the insight that our structure enable us to bring it to another level. So FGF21 is a hormone that manages energy utilization and helps the body adapt during times of privation. Adipose is the target for the acute glucose and insulin lowering effect of FGF21. The nervous system is the target of FGF21 and its effect on weight loss and reward behavior. So more information about it. So when you fast or starve, through activation of through fatty acid of a PPAR alpha, there's induction of FGF21 in the liver, plus different stresses such as high level of sugars, alcohol, induce expression of FGF21 in the liver, which then induces, sensitizes insulin in a dramatic way. It stops reproduction. When you're starving, it doesn't make sense for you to reproduce. You don't want to grow. And in fact, at very high levels, even the mice can be short. So, you know, it's really, really survival mode during times of privation. More information, if you're using recombinant FGF21, you can see that you can really induce weight loss. You can sensitize insulin. You can increase lifespan. This is not the first increase in lifespan described here. And it has this very fascinating reward behavior. And I would like to talk to Charles about that, about what it may mean in terms of neurobiology. But little more information. So this is basically the publication which showed that FGF21 regulates endocrine control of sweet and alcohol preferences by the liver. And basically this is what the conclusion. When you consume high levels of Coca-Cola or alcohol, there is induction of FGF21 in the liver, which then acts in the hypothalamus by really activating a complex of beta-Clotho FGF receptor 1C complex to really switch this, to really reduce this. Anyway, this is basically it. Okay, for some reason it went. So this is an interesting mechanism, and the question is, what does it really mean? Why Clotho has to do with all this function, and how does it work? Now, a little information about Clotho. Clotho was discovered quite a few years ago, more than 20 years ago, and it's a new gene which was shown to be involved in aging and basically a mechanism which involves a lot of metabolic processes, which was described quite a few years ago. Now, this is how Clotho looks like. It's a receptor, large extracellular domain, a signal sequence, and a transmembrane domain, and a very short tail. Now, what is really interesting here is that it involves two repeats which have similarity to glycosate hydrolysis. These are enzymes which really cut sugar. And it was thought, the current model is that Clotho proteins function as co-receptors for FGF receptor activation by endocrine FGF, similar to the role played by heparin. So this was the common model. And so we decided to have a look on it by structure, and we solved 4 structures. Structure of domain 1 of the extracellular domain at 1.7 angstrom, structure of free extracellular domain at 2. And structure of occupied FGF21 and FGF19, which I'm not going to show you. So again, like the similar experience you had, these molecules did not, we can make complexes, but they would not crystallize unless you added nanobodies. And we really met the same person you described, the guy from Belgium, and we also found that different crystal forms require there was no crystallization at all, pure proteins, unless the nanobodies. We have good evidence it has to do in inter-domain flexibility and crystal packing, but we got very high resolution. And this was a very difficult project. Many people tried to do it for years. And this is how this extracellular domain looks like. And this is the nanobodies that you see here. There are 2 repeats, and without any errors expected, they're really similar to sugar-cutting enzymes. They're very, very similar. So what happened here? A little background about glycoside hydrolyzers. So normally, glycoside hydrolyzers have 2 glutamic acids in their catalytic domain, which acts in concert to interact with hydroxyl here and really involve in the reaction. And this is how this takes place. One of the glutamic acid provides a nucleophilic residue, and the second one acid-base catalysis, and they act in concert to really release here, to cut the sugar. Now, it turned out that each of these domains of Clotho has only 1 glutamic acid. So domain 1 has a glutamic acid, only one of them, nucleophilic residue, and it misses, and so each of them cannot function in an enzyme, but they maintain the capacity of being able to bind to sugar, but they cannot cleave the activity. So now how the crystal structure of the tail of FGF receptor with Clotho looks like. So this is the 2 domains. We need this nanobody for crystallization. It's very elongated structure encompassing the 2 domain, and I've animated it. From the beginning, and I'll basically show you something which is clearer here, from the beginning to the end, it's about 50 angstrom involving interaction with domain 1 and domain 2 and the linker. It's very elongated interaction. It's from one side to totally different one. Let's look on it in more details. So this is how the structure looks like. This is what we call domain 1. And this is what we call domain 2. So I'll look in great details about this site. So this site occurs on an area which is nothing with the hydrolyze, with the sugar cutting activity. It involves multiple turns, which basically expose a hydrophobic surface on the ligand, which interacts with the hydrophobic surface in the receptor. And actually I have involving all entirely intramolecular hydrogen bonds. And this is basically the different turns which are color-coded to show you the interactions between them. So basically, this has nothing to do with the enzymatic, with the catalytic activity. It involves interaction with one site. And this is what we call site 1. And this is the second site, which is very interesting, because here the hormone is binding exactly to the catalytic pseudosite, which is inactive, of course. And what is really interesting is that there is a serine, proline-serine motif here, which interacts with the glutamic acid, which has been used in the hydrolysis, if this will be an active enzyme. So it provides a sugar mimetic. So a sugar-cutting enzyme evolved to become a hormone which regulates sugar by mimicking a site on the hormone which is sugar mimetic. And I'll show that to you in some details. So here you have a glycoside hydrolyzed from rice with a sugar. This is the sugar. It's really a rice protein, a rice enzyme, and this is the sugar. You can read what the sugar here. And this is the human receptor, hormone receptor binding to the receptor, and this is a very similar pocket. Let's now superimpose them. They're virtually identical. So this really shows you how a sugar-cutting enzyme evolved to become a receptor which regulates the level of sugar. We think this is quite potentially interesting in what's going on here. And so basically what I showed you earlier, that the normal enzyme uses 2 glutamic acids to cut the sugar, while they disappeared in Clotho, but it has still the capacity to use this site to recognize a serine, proline-serine motif, which is a sugar mimetic. And this is basically shown in this slide. This is the normal enzyme, which binds oligosaccharide, acts in ways to cut it, and this is the interestingly, even the hydrophobic side of the sugar are recognized by proline, which is sort of hydrophobic. So it's really maintained the site. So when I got this message, I read this: nature's imagination is so much greater than man's, she is never going to act as, by Richard Feynman. This is, I never would have thought, you know, it was very well known that these are evolved from enzyme to cut sugar, but now the significance is starting to emerge. Now, in addition, there's very good evidence that Clotho is associated with alcohol drinking, and as a gene product is necessary for FGF and regulation of alcohol. So there's something interesting hidden in the nature of this molecule. And basically, this is some SNP analysis which shows the link of alcoholism with Clotho. So there is interesting link there between this molecule and metabolism and also being involved. And this is again what I showed you earlier, that the molecules which regulate this process have these interesting responses in a withdrawal mechanism. And I just sum up to you, what do we know? We know the Clotho proteins contain a pseudo-glycosidase pocket. I didn't show you the result, but sugar derivatives can bind to this site. I showed you that FGF controls carbohydrate preference, and it's a major metabolic regulatory. And we do not think that this is a coincidence. We think that something interesting is hidden there in a way of these interactions. And also, now that we know that this is a pocket, in some diseases you want to block the site, we can use sugar derivatives or small molecules to develop new drugs. Now, in any event, this structure, so the mechanism of this family was not known, so this structure established the mechanism of how FGF and endocrine FGF work, and it demonstrated how this process takes place. And there was a lot, there are thousands of papers in that field, but no one really established the analysis. So we decided to analyze this mechanism in detail. After we solved the structure, we made the recombinant proteins and we studied the affinity. So it was proposed that Clotho is the core receptor, but actually you can see that FGF21 binds with very high affinity to Clotho. This is the key receptor. And then it binds extremely weakly to FGF receptor. But then this is offset by FGF interactions with Clotho. So you have here a complex where weak interactions lead up to formation. So based on that, we proposed the following mechanism, that there is an equilibrium between FGF receptor and Clotho, and that when the tail binds, this is the major site, and rather not the secondary site, and this leads to such a process of canonical ligand receptor activation. If that's really the case, and the function of that then is to just dimerize, maybe we can cheat the system, and it's chimeric receptor approach, which I've been using for many years. So this is Clotho here, and this is FGF receptor. Let's borrow and create, if they combine them together, what happens. And indeed, when you create such a molecule, and you bridge it with the nanobodies, which are dimeric, you can activate the receptor. So this is the mechanism of how it works. So these experiments demonstrate that rather than serving as a cofactor for FGF activation, Clotho functions as a primary receptor for FGF21 binding, and FGF receptor function as a catalytic subunit that mediates intracellular signaling. And it's not new, actually. This mechanism is similar to the mode of action of GDNF, in which binds to a ligand binding receptor and an RTK which doesn't have. So this is really known. And therefore, the title of our manuscript was "The Structure of Beta-Clotho Reveals a Zip Code-like Mechanism for Endocrine FGF Signaling." Basically, FGF receptors are expressed everywhere, and they can interact with, but how do you really decide that they become a target for Clotho? Based on where, for endocrine FGF, based on the place where Clotho is expressed. So if beta-Clotho is expressed here, you will have a response, and this is basically provides a zip code, and then it utilizes the canonical mechanism which is activated by these receptors. So this is why we think that, and so the mechanism which was described based on these many studies is not. So now there's an interesting link to cancer. So this mechanism can really become somehow rearranged in different cancer, and interestingly in liver cancer, in a subtype of liver cancer. So the beta-Clotho FGF receptor pathway is activated in hepatocellular carcinoma by aberrant expression of FGF19 through an autocrine and paracrine mechanism. And a substantial population of liver cancer can benefit from such a treatment. And these are all in humans. This is not in any animal model. And basically, I'll remind you how that happened. I showed you earlier that FGF19 is normally expressed in the intestine and it acts to the liver to regulate these processes. Okay? And it acts on liver to stimulate this process. FGF21, on the other hand, is made in the liver and it acts in the hypothalamus to regulate these processes. Now, what happens in particular subtype of liver cancer is that they aberrantly express in the liver FGF19. And then it acts on FGF receptor 4, which is the mediator of cancer. So there is reprogramming. And if you look on the Genome Project, actively elements of this process are amplified in 5% FGF19, 5% FGF receptor 4%, and beta-Clotho 3%, mutually exclusive. So it's about 10 or 12 or 15% of liver cancer, human liver cancer, which can benefit from this. And basically, we really try to understand how aberrant expression of FGF19 in the liver can really cause cancer, where FGF21 is all the time there. It's made there at very highest level. And we don't know yet the answer, but I can give you some information. Again, we were looking for quantitative data. We can really show actually that interestingly, FGF19 binds to FGF receptor much more weakly to FGF1C. It's like tenfold, and yet it's much more potent stimulator. And the interactions here are very similar. So it must be something to do with the kinase activity, with the dynamic nature, which we are still exploring, because this pair leads to robust mutagenic response, and this stimulation is not mutagenic at all. So there is interesting question there to address. So the affinity of the ligands to the receptor is really very, very weak. So therapeutic approaches that we are developing for treating liver cancers are several of them. Antibodies that bind to site 1 and site 2, novel engineered inhibitory ligands. So in some diseases you want to block these pathways, but in other diseases you want to stimulate these pathways. So there's a variety of new opportunities here for drug disease. For example, agonists, new potent engineers, endocrine FGF, can be used for treating metabolic diseases such as NASH, pancreatitis, and I'll show you the evidence, and there's quite a lot of clinical trials. And in this particular case, you want to generate agonists and stronger. So for cancer, you want to create antagonists, but for metabolic diseases and all that, you want to create agonists. And you're just looking into this. These are the different diseases which you may really benefit, and there's a lot of activity in the industry about this process. But I'll show you one experiment. Just by looking at the sequences, we can really generate already a potent FGF21, which is about tenfold more potent in binding affinity and stimulation of kinase activity. And using various expression systems, we can now really make very potent. And you can see here a list of the clinical trials which are now ongoing for NASH with different, and there are two forms of FGF29 which are pegylated, which seems to be quite encouraging. So the idea is to use our super agonists also to treat, and this is something which is ongoing. So, in summary, in the second part, I gave you information about canonical and endocrine FGFs and capturing and activated states of FGF receptor. And I showed you the surprising result that evolution of a sugar-cutting enzyme to become a receptor for metabolic hormone and a zip code-like mechanism for endocrine FGF signaling. And finally, new opportunities for drug discovery. This part of the work was done in my lab, including Eric Lax and Sunho Lee, who solved the structures, and many other members. And of course, this Jan and Els are the same people that develop nanobodies. Thank you very much for your attention.